flu or with the RNA viruses in general is that like the virus evolve or the virus change so quick. And so the vaccine that we use to vaccinate the animal this year may not be able to protect the animal next year, not because the animal do not have the immunity anymore, but because the new virus appear, right? And the immune system is basically like not stimulated to work with the new virus. So that is the main challenge for vaccine against influenza virus.
Swine it.
Hello, and welcome to the Swine It podcast. I'm your host, Tara Donovan, and I'd like to introduce our guest today, Dr. Hiep Vu. Dr. Vu is an associate professor at the University of Nebraska-Lincoln. Welcome to our show, Dr. Vu. How are you?
I'm very well. Thank you so much for having me.
Absolutely. So, Dr. Vu, could you introduce yourself to our audience and just give a quick history of your career path, how you came to University of Nebraska, and what you do today?
Yes, so my name is Hiep Vu, and I right now an associate professor at University of Nebraska-Lincoln, and I grew up in a swine farm, and so I help with my parents raising pigs and especially apply vaccine to pigs, and I saw many times we try to save the vaccine because it's expensive, right? And then it caught up with the disease. So I'm always curious, how could the vaccine protect the animal? And so I went to the vet school in Vietnam, learned about disease stuff, and didn't answer my question: how would the vaccine work, and how would it protect the animal, and how to design the vaccine? So I came to University of Nebraska-Lincoln 2007 to start my master degree, and I've been here since then. So I went through my master, my PhD, and postdoctoral associate, and then I became an assistant professor in 2017, and now I'm an associate professor. So my main focus still on swine vaccine development because that's my heart there.
Right. Goes back to your childhood days.
Yep.
So absolutely. Well, I grew up in Nebraska on a farm, and I also went to University of Nebraska-Lincoln for my undergraduate. So we have a little bit in common, and I love your shirt. So still a Husker at heart.
On purpose, yeah.
Great. Well, so I want to add, so Dr. Vu, you have a long history of looking at viruses and vaccinology at University of Nebraska. And so just for our audience, a team of scientists led by Dr. Vu and Dr. Osorio are recognized with winning the New Tech Ventures Breakthrough Invention of the Year in 2015 for their work with a promising vaccine against porcine reproductive and respiratory virus, we all know as PRRS. And so congratulations on this. And being a practitioner, having dealt with PRRS for years on a daily basis, I'm excited to hear that there's continued work and promise for a solution, hopefully sometime out there. That, vaccines haven't always worked as well as we'd like them to on some of these viruses that are able to change and mutate. And so that's exciting. Thank you for that.
Well, thank you so much. Yep.
A worldwide leader in animal nutrition, Adiceo's portfolio of products includes methionine, the full range of vitamins, enzymes, organic selenium, probiotics, mycotoxin management strategies, and palatability products. With such a diverse offering, Adiceo supports its customers with a broad range of expertise, tools, and services to help them maintain a competitive advantage. Adiceo, fueling predictable profits. To learn more, visit Adiceo at www.adiceo.com.
That exciting part of my project. So that was I would say thanks to that part of the research that make my career, my position right now, because I did that project when I about to done with my PhD and just starting my postdoctoral, and that was my first independent research project.
Yeah, so it's dear to your heart and helped pave your way to where you are today.
Yep.
Yeah, excellent. So instead of PRRS virus, we're going to talk about a different virus, but still in the swine area, and that's swine influenza. So pigs are still dealing with that every day out in the field, and so you've got some vaccine research that you've been working on, and that's what our focus is going to be on today. So can you just tell me about? Let's talk about swine influenza generally first, just so that we can all get in our minds a quick review.
Yes, so swine influenza is somewhat like similar to the seasonal human influenza because they are influenza anyway. So swine influenza is an RNA virus, and because RNA doesn't have the mechanism to correct the mistake, and so they have the tendency to mutate faster than when we compare to DNA viruses. And so the challenge with the flu or with the RNA viruses in general is that like the virus evolve or the virus change so quick. And so the vaccine that we use to vaccinate the animal this year may not be able to protect the animal next year, not because the animal do not have the immunity anymore, but because the new virus appear, right? And the immune system is basically like not stimulated to work with the new virus. So that is the main challenge for vaccine against influenza virus. And swine influenza virus is not much different than human influenza virus because it caused like mostly the respiratory disease.
Right, right. And so my understanding with vaccine technology for swine influenza is you're looking at a vaccine that's able to be recognized and attached to this outer capsule, right? These areas on the outside of the virus, and that's what basically is the recognition part, right?
Yep.
And so we name viruses or influenza viruses with an H and an N, right? Strains,
Yep.
using those outer capsule proteins, neuraminidase and hemagglutinin. Is that correct?
That's right. That's right.
Yeah. Yeah.
So for influenza virus, like you just said, like basically we name them based on the H and the N. And for swine influenza virus, mainly we have H1N1, H1N2, and H3N2. So those are the three subtype of virus that infecting swine most of the time.
Right. So tell me about your vaccine research. So dig in a little bit to what you're working on and how that might be different or the same as some of the vaccines we've used out in the field.
Okay, so I think like I go a little bit basic concept first, right? Because virus is something that's very interesting in the way that they must enter the cell to replicate. So if the virus cannot enter the cell, they cannot do anything. So because that the one way to design the vaccine for viruses is that to induce the antibody to prevent the viral particle from enter the cell. So in order for viral particle to enter the cell, they must interact with the cellular receptor. And for influenza virus, the hemagglutinin gene is the surface, and the viral hemagglutinin go bind to the receptor on the cell, and to facilitate the entry of the virus into the cell. So now, most of the time that people target is that induce the antibody against the hemagglutinin protein, and so that to break the interaction between the viral particle and the cell. So I think like I should use an example of COVID for because I hope that it easier to understand because for COVID, then the spike protein is the one that allowed the SARS-CoV-2 to enter the cell. So all of the vaccine company, regardless whether Moderna, Pfizer, or like Johnson and Johnson, all of them target the same spike protein because the idea is that when you produce the antibody against the surface of the viral particle, the antibody will prevent the viral particle from enter the cell. So for flu, come back to the flu, then the hemagglutinin is the target. Now you know the target, then you need to deliver that into the animal. So different company, different research group have different way to do so. The most traditional way is that like we take the piece of gene and code for the HA for the hemagglutinin, and we use the protein expression system like baculovirus or some protein expression system, and you express you purify the protein. Then you have the protein, you formulate with adjuvant, and you inject to the pig, and the pig will make antibody. So in that case, that means that you have to go through all of the process to make the protein and purifying that. The other technology that we have is that instead of making the protein, we make the messenger RNA, right? So from the HA, we make the messenger RNA, and then we inject the animal with the messenger RNA. And then messenger RNA, when it goes to the animal, it will be converted into protein. And the protein is the one that stimulates immune response. The messenger RNA by itself won't be able to induce protection. So that worked very well for the SARS-CoV-2, as we know so far, right? The Moderna and Pfizer vaccine is based on the mRNA encoding for the spike protein. That technology can be applied for swine flu as well. The only challenge is that it's expensive. So when we make the vaccine for swine, we cannot charge like the producer $30 per dose. We cannot ask producer that you have to have the -80 freezer to store the vaccine, because mRNA have the tendency to degrade quicker. So with that in mind, then I came up with the approach that we use the DNA plasmid instead of messenger RNA. So the DNA plasmid is a piece of DNA where you have the hemagglutinin gene in there, and you can produce the plasmid in E. coli, in the bacteria, in the BSL1 condition. The advantage of the DNA plasmid is that it is very, very stable. You can store the DNA plasmid at room temperature for a while, and then no degradation. In the past, people had used the DNA vaccine a lot. So basically, the idea that you inject the DNA to the animal, and then the DNA will be enter the cell, just like the messenger RNA. Once it enter the cell, it will be converted into the messenger RNA, and then the messenger RNA will be converted into the protein. And again, protein is the only molecule that stimulates the immune response. It stimulates the production of the antibody or the T cell. The DNA or mRNA won't be able to do so. It have to become protein is the key. So people have used DNA vaccine for many years, but the efficacy is very low. The reason is that the DNA molecule cannot enter the cell effectively. If you just simply inject the DNA plasmid, it do not enter the cell very well. And if the DNA plasmid does not enter the cell, they cannot be converted into protein, and they cannot induce the immunity. So with that in mind, what I'm doing is that I have the DNA plasmid like usual, but I use the what we call lipid nanoparticle. Basically, we wrap around the DNA molecule with lipid, and so we call the lipid nanoparticle DNA vaccine. The main purpose is that the lipid nanoparticle facilitate the entry of the plasmid into the cell, and so that the plasmid will be converted into protein. So that is the innovative is there. By the way, when I mentioned the lipid nanoparticle, I need to mention it that the messenger RNA, the Moderna, the Pfizer vaccine, they also wrap it around with the lipid nanoparticle, because the messenger RNA molecule by itself do not enter the animal cell, the human cell, effectively. So the key breakthrough or the key component to make the messenger RNA work well is because they found the lipid formulation to encapsulate the messenger RNA. So um, I do the kind of similar approach. I try different formulation, and I found one formulation that worked very well. So when we encapsulate the DNA plasmid with the lipid nanoparticle, it worked very well. So I had been demonstrating that one dose only. We just need to inoculate. We just need to inject the animal with one dose, and it induced very good antibody response.
Wow, that sounds great with one dose.
Mhm.
So a lot of steps to that, it sounds like, but a lot of the steps are already been used in vaccine technology, in the past for either other viruses, and so it's really, it sounds like to me your work as incorporating a lot of those pieces together for swine influenza, and then working out the very last step, which is that lipid nanoparticle, and what that looks like, because it sounds like that must be different for different viruses, that particular piece of the vaccine.
Not necessarily like the lipid nanoparticle. We call it like more as the delivery mechanism.
Okay.
right? So the lipid nanoparticle, it quite flexible. Like if I want to, like I can use the same approach, and let's say like I make the DNA vaccine for PCV2, for example. It won't be like so different. Um, Actually, like because when we come to mRNA, people sometimes like misunderstood that like we just started to make mRNA vaccine since the pandemic came. It's not correct. People already researched on mRNA vaccine for many years, and then they apply that to make the mRNA vaccine for the COVID. So research it have the process. It generally like build upon one the other, and so the DNA vaccine have been used for many years with limitation, and we improve it by the lipid nanoparticle.
So tell me about influenza then with this vaccine technology. Do you have to change the vaccine as this virus changes out in the industry, and how is that done?
Yep. So you asked the right. I mean, the heart of the problem is that the influenza virus changing all the time, and so there are two approach for the vaccine against influenza virus. One approach is that like, you design the vaccine in the way that like, you make it one time only, and then it will work against every possible um, influenza virus out there, like regardless of how long after the time you make the vaccine. And in that case, we call the universal vaccine or the broadly protective vaccine. That one is difficult to make, and the other approach that people are doing, and especially like in the field of swine veterinary people are using, is that we change the vaccine when the virus change. Because nowadays with the sequencing technology and with the surveillance that we have, like we kind of monitor the change of the viral sequence very like real timely, and so we update the vaccine whenever there is new virus appear. In order to do so, we need to fulfill certain requirement of USDA to license such a vaccine, because the traditional approach is that you grow the virus up and then you inactivate the virus and you mix with adjuvant, and we use the inactivated vaccine. And with our approach, with that current approach, it takes a long time to update the vaccine. So USDA had the new regulation they call the vaccine platform. So if you use the non-infectious non-infectious vaccine, so the DNA plasmid is non-infectious, and so when it's non-infectious, you inject the DNA, or like if you accidentally like release the vaccine vial in the farm, you don't worry too much about like it become a virus and it spread around, so it's safer. So USDA had the new mechanism that if you use the non-replicating, non-viable vaccine, then you just need to went through the licensing process. You still have to went through the licensing process, but once you license it and you have the standard protocol, you don't change anything. So when there is the new variants of flu come, you just swap the gene, and that the DNA vaccine is perfect fit for that purpose, because it's very easy to construct new plasmid. And so when there is the new virus come, I can quickly ask the company to synthesize for me the new plasmid, and I can update that. So my goal is that like not to generate the vaccine that protect against every possible influenza. Instead, like I try to make the vaccine in the way that I can update quicker. So that is the advantage of the DNA platform that I am pursuing.
Okay. And so that plasmid, though, is still going to be targeted to the hemagglutinin piece of the virus. So there will be some changing that has to occur as that virus evolves in the industry year to year. But from what I heard, you're able to adapt to that because it's a platform and it's not a live vaccine. It's a safe vaccine, so it can be done by using some of the surveillance that we use in the industry.
Yes, and beside like using update the vaccine, the other thing is that like with the swine especially, like we had very large operation, right? And so one operation will have a unique set of influenza virus affecting their farm compared to the other. And so like I can custom make the vaccine for that particular farm, because the large volume that they acquire. It may not work for human because you cannot make one dose of vaccine at the time. But swine, right, one operation they may have like 10 of thousands or more. And so one batch of vaccine, I can custom make the vaccine for that particular farm because I know the sequence of the age of the influenza virus that affecting that farm. So that can be used more like the prescription vaccine.
So, what are the next steps for your vaccine?
So right now, we demonstrate in the experimental condition, it worked very well. Like one dose of vaccine induced protective immunity. Like for flu, because antibody is the reliable parameter of protection, and we saw that by 14 days after we applied the vaccine, we had very high antibody titer. And so I guess that like the vaccine would mediate protection by about 14 days after vaccination. We don't need to wait very long. And because one dose only, then it shortened the time. I remember when we used the mRNA vaccine for COVID, 2 doses and 3 weeks apart. And so from the first time we get the shot until we are immune in about 5 weeks, because you wait for 3 weeks to take the second dose and 2 more weeks to have the time to induce the protective immunity. So it's 5 weeks long. So by one dose, it reduced the time to like maybe 2 weeks, 3 weeks, shorten the window. So that's what I demonstrate in the lab right now. But the next step would be like to do the dose titration, because right now I do only one single dose and I use a little bit higher dose of plasmid. And so my next immediate experiment would be like to do the titration to determine the lower possible dose of plasmid that induce protection. The other thing is that because we had multiple subtype of flu co-circulating, so one farm potentially can be infected by two different subtype of influenza virus. So right now, what I demonstrate that the vaccine is monovalent. So I use only one HA gene. So the next step is that I want to incorporate like multiple HA to make a polyvalent vaccine, and that will be like a part of research that I need to conduct to see like what is the minimal dose, and how about if we combine like two or three different HA gene into one single vaccine, how does it work? And another thing is that for flu, when let's say we vaccinate the sow, and the sow would transfer the antibody to the piglet, if it exposed to a heterologous flu, then the antibody not protect the piglets, but potentially like enhance the pneumonia. So the antibody may not help but hurt because it enhances the infection. So I want to see whether the lipid nanoparticle DNA vaccine can avoid that type of enhancement or not. And also for flu, if the piglets had maternal antibody, that antibody potentially interfere with the vaccine efficacy. So we want to know like whether our DNA vaccine kind of bypass the effect of the maternal antibody. So there are quite many outstanding questions I need to answer, right? Because anyway, I am in the very beginning of the project. I just started it a little bit over 1 year ago.
Okay, yeah, you touched on all of the questions I had regarding having used influenza vaccine in the field for several years. One that I have that you didn't cover is my experience is that the immunity from a killed product given as a 2-dose, the immunity, the duration is quite short. And so do you know anything about that with your vaccine technology, or is that also on the list of things that you need to discover?
Well, that is on the list as well, because I just like started about over a year ago, and right now, like my main focus is to find the right lipid nanoparticle formulation to effectively deliver the DNA, and we found one right now, and so we are moving forward with that. So I'd like to reduce the dose of the vaccine down, and then I also want to see like I can combine multiple HA, and then down the road, like I like to study the influence of maternal antibody, and then obviously the duration of the immunity. But I think the duration of immunity may not be as critical as in human because the commercial life of a pig is very short. Perhaps the breeding stock may need a longer duration. So um, that is one more question to the list.
Yeah, absolutely. Well, some exciting things though, and continuing to work on vaccine technology is so important because unfortunately we keep seeing new viruses showing up and or their ability to, it seems like, become more of a problem or more virulent. So I appreciate that work in all of the swine viruses that you've been working in.
Thank you.
So, with that kind of new introduction, I have a quick question. I know this is kind of hot off the press, but there's been a syndrome in dairy cattle in the South in the U.S. in Texas and Kansas and New Mexico, and the USDA has announced that they have some proof that it could be high pathogenic avian influenza that's causing this syndrome. So, as a virologist working with an influenza virus, what do you think about that? And will that have some implications on, swine? Could that be an issue?
Yes, so I saw the notification last night, but I haven't been able to read the detail yet. One thing I know is that the swine is susceptible to avian flu because people use the term mixing vessel to describe swine. Swine can be infected with the seasonal human flu, and actually, like human, especially the farm worker transmit the um, the seasonal human flu to swine very often. But then swine also been potentially susceptible to the avian flu, so they can potentially create the mixed virus or we call the reassortant virus. And actually, the 2009 pandemic flu it carried a gene segment of avian and then classical swine and some gene segment of human influenza virus. So yes, swine is a susceptible species for avian influenza virus. But the news is that the cattle is also susceptible. Is a little bit tricky somehow. In my I try to find the word. Yeah, it's substantial because we do not think that cattle is susceptible to avian flu. Now, if it is susceptible, then it is a bigger concern. Actually, if you follow the news, you may notice that I believe early this year or late last year, there is an outbreak of Hiep flu in sea mammal as well in South America. So it have the news, according to what I follow, it have the tendency to think that the avian influenza virus is kind of expanding the host range, it infecting more and more species, and it's a concern.
Yeah. So, would your vaccine technology be able to be used in cattle? Say this progresses and they need a vaccine. Is it adaptable to other species?
Yep, that is the goal because I try to develop more like a platform rather than like one particular vaccine because, like I mentioned in at the very beginning, viruses must enter the cell to replicate, and depend on viruses. But like some of them use only one viral protein to attach to the receptor. Once we know that protein, we can clone it into the plasmid and then wrap it lipid nanoparticle and vaccinate the animal. So I try to make this approach become the platform, and when there is new viruses come, especially with the virus that we know the receptor binding protein, then we can quickly adapt the technology. So it would not work for some virus where like you don't even know which viral protein is required to infect the cell. In that case, it's difficult. But for this new avian flu, I think we can adopt this technology and use in cattle.
Well, that's great, and to me, really intriguing, or maybe I don't know the right word, scary, in that cattle haven't been susceptible to influenza A, the avian influenzas previously, at least that I'm aware of, and so this is really interesting news, and hopefully we can come up with solutions like yours to help these ever changing viruses.
Yep. So flu is one of the complicated viruses because they infect it infect multiple different. It will have very wide host range, and it might somehow. I always think that if any viruses that have the wildlife species involved, it's very hard to control because you can apply the vaccine to every single people on Earth, every pigs on Earth, but like how can we vaccinate the wild bird, the wildlife animal, right? And as long as we stop vaccinate the pigs or something, the virus will come back. So to me, like it if it involve wildlife animal, it will be difficult to control.
Yeah, it's going to be a challenge, that's for sure. So we'll learn more, I'm sure, in the coming
Mm-hmm.
months of
Mm-hmm.
if this is true and what that impact will have on the rest of the food animals and particularly swine for us. So
Yep. Yep.
It's time for our famous three. There's a better way to finish your pigs, and it's called Digesteron Finish. Digesteron Finish provides higher feed acceptance, leading to improved digestibility, so you get a more efficient finish. Made with all natural ingredients, Digesteron Finish is a three-way win for producers through better animal well-being, enhanced economic performance, and improved sustainability. Digesteron Finish can help you get more pounds in less time, so why wait for your finish? Learn more at dsm.com/finish.
well, great. Thank you so much for sharing your vaccine technology with us. I have just a few more questions for you. With all of our hosts, we ask a few questions just to get to know you a little bit better and for us to maybe get some added resources that we can go check out and learn more. So the three questions that we have, the first one is, what is your favorite book or resource that you use in your area of expertise? And so my guests always jump to a book, but it doesn't have to be. It could be a research group. It could be a meeting. So that type of resource as well. But what do you use? What's the one thing that you reach to stay current or to maybe go back and learn the fundamentals again?
So in my um, in my field, like there are two conferences that I really like to go every year. The first conference is we call it North American Pest Symposium, but now it is changed to the International Swine Viral Disease Symposium, and that that is happen every year in Chicago, the first week of December, and that's what I've been attending like the past 16, 17 years. So that's where I'm gonna meet with my peers, as well as with swine practitioner, producer potentially, and learn more about the current problem in the field because I I more like work on the lab that develop vaccine testing under experimental condition, and I want to hear what is going on in the field right now. The second conference that I normally try to come is the American Society for Virology, and that is a very, very large conference, and it had very deep or cutting edge technology and all the new knowledge people will share. And so those are the two conferences that I like to attend, possibly like every year.
Yeah, absolutely. I have attended when it was the North American PERS symposium before, and I
Mm-hmm.
can tell you, I'm always humbled when I sit in that chair because I don't know all the very small detail like you do about virology, but it always makes me happy that there's work and really smart people working on vaccine technology to help us in the field, but also that there's so much an area for different researchers to share and then for us to interact. That's really the only time I interact with researchers like yourself is that conference.
Well, the North American Pest Symposium is an interesting part because you're gonna hear a lot of basic science, but at the same time you're gonna hear from the practitioner and from the industry standpoint. So I like that conference because like I got exposed to things that I normally daily I don't see it. So I love to chat to practitioner and learn more about the problems.
Yeah, absolutely. So what about your favorite book or resource outside of virology, outside of your field of work?
Well, I like to read some what's say like not really fiction, but like I read books about, well, still related to my field. I read book about my infectious disease, and some of them are very exciting to read as well. And one of the book I like a lot is it called Spillover, and they describe like because in many pandemics it have the origin from animal. Okay, so whether we like it or not, but that is what I when I teach a student, I say like we divide like biomedical, or we divide like human medicine and veterinary medicine, but the virus really do not care. Virus do not care whether that is animal or it in a human. It's just a host. So the spillover is the book that it very interesting to read, and so we understand like because the interaction between humans and animal, then uh, cross species, for example, the animal virus from time to time come and infect humans, and because it jumps species, like basically everybody are naive to that new viruses, and the virus will spread tendency to spread quick, and it create the pandemic. So spillover is the book that I really enjoy, and the other one is we call the hot zone. We talk about the Ebola virus, and that is also another book. It's a little bit scary to read, but at the same time, it's exciting as well.
Yeah, I have. I think Hot Zone was maybe made into a movie as well. Um,
Mm-hmm.
so yeah, that we just talked about swine influenza and it being in wildlife, which is really going to be challenging. And the spillover, right? It's real. It's what we're living with right now. So
Well, in the spillover, they mentioned like, for example, like at a vet, like when you practice and then your client call you maybe like 6 7 p.m. and you rush to see the animal, and sometimes like the very simple thing, like for example, they didn't wear the glove when they examine the animal, and potentially like they may contract the pathogen. So I remind my student like all the time that like, well, be careful, because we don't know what we are examining, and so always think about safety first. But in the spillover, it's a very great book to read. And then like if you think about vaccine, I think that the other book that I like to read or introduce is called um, vaccines. The title it talk about, cannot say his name. He's work from Merck, and he developed just so many vaccines. I don't think anybody else on earth can make more vaccine than him.
interesting. So it sounds like you are a hard worker. You read for pleasure. Some of the things that still go into your work, and that's maybe true for a lot of us. Those
Mhm.
types of books really, for me, are interesting as well.
Well, that is perhaps like it kind of fuel. Like it make me more curious and to ask question and to read. But yeah, those are the author is so awesome in describing. I sometimes like think like I cannot write it. I mean, I know about it, but like when I read their writing, I say, wow, they explain this so easy to understand, but at the same time, make it very exciting to read too.
Yeah, absolutely. Well, thank you for sharing those. So the last question that I want to hear from you is share an attribute or characteristic that you feel sets successful professionals apart from others. So what's an attribute that you look for or that you highly regard?
I think like my, I think the focus is the key. The focus not meaning that we focus on this moment, just do this type of work, but like focus on achieving the goal. So we need to identify the big problem to solve, and then like focus our determine like to re- to achieve that goal. And sometimes in the field, like sometimes it 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 easily be distracted by other things, because for example when the SARS-CoV came, like basically like obviously the public is fear, but the scientists, like, I wouldn't say exciting, but like that is a new chance, like new viruses and a lot of more funding, and sometimes like we may shift land like we and change to work on the other things. So I think like I like to stay focused, stay on my lane, try to solve the problem that I set to solve. I think that helped, and also Also, like I said, I grew up on the swine farm, so I like to design something that more practical to the producer, because there are times I'm thinking about solution or vaccine type products, but then later I realize, well, nobody gonna really, it not practical for swine purposes. Because an example I've been saying to my student is that if we make the vaccine and we ask the swine producer like do intravenous inoculation, it's not gonna work. Or like, so that in an example. So yeah, stay connected with the producer and really understand what is the key problem to solve.
Absolutely. And I appreciate that because I have been, educated on technologies that I think are really new and intriguing. But as a practitioner, I can sit there and say, I don't think that's applicable to what we can actually get done in the field. And so I appreciate your end goal in mind as you're working in a really technical field, that, going back to your parents' farm and saying, could my parents really be able to use this on the farm? I think that's great, and a really a good thing to share with our listeners to think through the entire process.
Well, I always like, I go to conference, I learn new technology, and I love that. But then like I keep it in my perhaps toolbox, and I try to see until when it appropriate, or like whether it appropriate. I don't mean that like it not never be used, but like I just keep it there as resources, and then like try to see maybe it not feasible at this moment, but like when the technology evolve, potentially one day become like applicable.
Absolutely. Well, great. Thank you so much for your time today. Again, this is Dr. Hiep Vu. He's with the University of Nebraska. And if you have comments or questions on your platform, just send them out to us. If you liked our show today, give us a like. And I appreciate your time today. Thanks so much.
Well, thank you so much. It's a great pleasure talking to you today.